So this is lab 5, where we're going to quantify the amount of bacteria in a sample. Specifically, this is the soil sample. So you're going to have to get soil from around your house or anywhere else that you would like. So before lab, you're going to do all the things that you're supposed to do, like disinfect surfaces with bleach or isopropyl alcohol, wash your hands, and of course wear all the appropriate PPE. And then also you're going to need a soil sample from outside. The amount you're going to be using is about 1 teaspoon, so you don't need a lot.
Okay, micro students, so this is lab 5, where we're going to be quantifying the number of microorganisms in a soil sample. So you're going to need a variety of things. I have my soil sample. I have 6 plates that I've already poured. I have already labeled them. Remember, we label on the bottom of plates. You're going to need, these are called conical vials because they are cone shaped at the bottom. So these are 15 milliliters. So we'll need 6 of those. The phosphate buffered saline is the solution you're going to need. And then some, the pipette pump along with the 2 ml pipettes. And then these are called spreaders. I don't think we've used these yet, but we're going to use these in this experiment. Okay, so I also have my tubes labeled, and so the first thing that we're going to want to do is get some soil into our tube that's labeled as stock. And so I'm going to do the best to pour this in here without spilling. So this is just from my front yard. Okay.
So you want to add about 1 teaspoon or 5 milliliters of soil.
All right, so even though I made a mess, there is our soil in a tube. And then I'm gonna add our phosphate buffered saline. We're gonna add 10 milliliters. Okay, so there's 10 milliliters, and we're going to add that to our soil. So make sure the cap's on well. We're going to give this a nice mix. Make sure all of that's suspended. And then we're going to let that settle. I've already done the first step, which is to take some of my soil and add it to this vial. And then I added 10 milliliters of this phosphate buffered saline to it. So you use your graduated cylinder, and you want to be as precise as you can to get 10 milliliters. So use the graduated cylinder, not the markings on here, because these are not as accurate. So we want to be as accurate and precise as we can. And so that's the point of using this graduated cylinder. So fill this up to 10 milliliters and add it to this tube. And I've given it a little shake to really mix the soil in with that phosphate buffered saline. So the next step we're going to make it's called a serial dilution. And so what that means is we're going to take a sample and from one and dilute into the next in a series. So we're going to go from the first tube to the second, and then mix that, and then from the second to the third, and so on and so forth. And so I have pre-labeled these tubes. They have, it says 10 with an exponent on it. That says -1. This one says -2. And so those represent how much we've diluted the original sample. And so we'll talk more about that later. But I'm going to glove up. And then what I'm going to do, and I'll probably edit this video to show this, but I'll do the first one. We want 9 milliliters of the PBS, that's phosphate buffered saline, into each of these tubes. So I'll do the first one, but you probably are not going to want to watch me do all of them. So again, you want to be as precise as you can. So 9 milliliters exactly. So that's 9 milliliters. And that's going to go into my first tube. And then I'm going to do that for the remaining tubes. So I have added 9 milliliters to each of my tubes. And so now what we want to do is continue with that serial dilution that I was talking about. So our first tube, this has had a chance to settle now a little bit, and we're going to use one of these sterile pipettes to transfer exactly 1 milliliter from our first tube So I'm going to draw up 1 milliliter. And so this is our soil tube. This is our first tube. It should be labeled 10 to the minus 1. And I'm going to add all of that milliliter. And then I'm going to continue to use this. I have the cap on here. I'm just going to give this a little shake to make sure it's well mixed, because now I want to take out of this 1 milliliter. And then we're going to go to, now we're to the third tube. This should say 10 to the minus 2. So I'm going to add the milliliter of this, cap it, and mix this one. And now I'm going to take a milliliter out of this one. So this is our 10 to the minus 3 tube. Give this a little mix. Draw up 1 milliliter. Again, you want to be as precise as you can, so it's right on that 1 mil mark, because this will help us in our calculations later. So I just added to the 10 to the minus 4, mixing, draw up 1 milliliter. And then the last tube is labeled 10 to the minus 5. And I'm going to add to that. So what we've done, let me just cap that. I'm going to give this a mix too. So we're done with this pipette. So I'm going to put that aside. We've made a series of dilutions where this would be full strength bacterial concentration. But now we've taken 1 milliliter of that and added it to 9 milliliters of this sterile phosphate buffered saline. And so what we have effectively done is what we call a 1 to 10 dilution. So we've diluted the number of bacteria from our soil exactly 10 times. And that's what that 10 to the minus 1 means. It means 1/10. So this is going to have 1/10 the bacteria as our original. But then we repeated that. We took 1 milliliter out of here and put it in another 9 mils. So the total volume is going to be 10 mils. So another 10-fold dilution. So this is now 100 times less. This is going to be 1,000 times less. This one's 10,000 times less. And our 10 to the minus 5 is 100,000 times less. So now what we want to do is take each of these dilutions and put them onto the corresponding plate. So I have a plate here that says stock, and so I want to take from my stock tube and put onto my stock plate. And so the way that we're going to do that is again using our sterile pipettes, but then we're also going to need a spreader for this. So what we want to do is add only 0.1 milliliter, roughly about 4 drops from each tube onto the plate. Okay, so that's what we're going to do. And you're going to use a fresh pipette for each of these plating. So this is my stock. So I'm going to draw up So unfortunately, there's not a line that you can use for 0.1 because the smallest is 0.2. So this first line is 0.2. So you just have to do the best you can to estimate. that's good. So we're done with this one. And then you don't want to just leave those drops there, but what you want to do is use the sterile spreader. And so don't touch that end. So again, lift the lid, and then you're just going to take that liquid and smear it around or spread it around using the sterile spreader so it's completely covers that surface. Okay, so this one is done. Now we want to do the same thing and repeat for all these remaining. And so I'll do one more. This is my 10 to the minus 1 tube, and this is my 10 to the minus 1 plate. And so I'm going to do a very similar procedure where I'm going to draw up some of this sample. So again, we mixed these previously, but it doesn't hurt to mix it again before. And so I'm going to draw up this liquid. And then I want to dispense 1 milliliter, I'm sorry, 0.1 milliliter on there. And then using the fresh sterile spreader, we're going to spread. So again, when you lift that up, try to keep the lid and then spread it. I'm kind of rotating the plate as I go. Just so it's well spread. And then that's our second one. So I'm going to repeat this for the remaining 4 tubes and plates using a fresh pipette and a fresh spreader for each transfer. So I've plated out now each of these 6 plates coming from each of the corresponding 6 dilutions that I have. So now the only thing left to do is to parafilm these and set them to incubate like we've done in previous experiments. So that's what I'm going to do. And then next time I show you these, it will have some number of days growth on it to hopefully see some bacterial colonies forming.
After lab, you do want to parafilm the plates as we always do when we incubate, but then also remember to photograph your plates with your ID. And then as we finish all labs, we want to disinfect the surface that you are working on and wash your hands.